Welcome everyone to another exciting episode of Sourav Bhattacharjee Show. In this episode, I chatted with my colleague in UCD School of Veterinary Medicine, who is a globally renowned expert in tuberculosis, which is again having a resurgence and wreaking havoc throughout different continents in the world. I had an exciting chat, slightly technical, but still very interesting, where Professor Stephen Gordon has spilled it all with all his understandings and pearls of wisdom. This is a very exciting episode on a topic that is pertinent and that is clinically hyper-relevant. Sit back, relax, and enjoy my chat with Professor Steve Gordon. Welcome, Steve. Thanks for coming to my show, and I'm really, really honored that you are here. Now, as I always say, that it feels kind of bizarre that we meet all the time and bump into each other into UCD School of Veterinary Medicine, where we both are working. But at the same time, it's kind of have a kind of a more formal kind of an ambience where I meet up with you. Again, it's a nice opportunity to kind of have a more esoteric discussion, probably.
Yeah.
And if you are here, the topic has to be about tuberculosis, which you are, as I must say, you are a globally renowned and respected expert on this. But before going to all these scientific terms and scientific discussions, I always try to understand my guests on where they started their trajectory, because they are not only established researchers, but they are also kind of role model or kind of a good example for the upcoming researchers. So give us a brisk tour about your career and how did you get into this field of tuberculosis?
Well, first of all, thanks very much, Sourav, for inviting me here. So my journey in tuberculosis, it's a long one. I've been working on tuberculosis actually since around 1991. And I was originally interested
I was in school then.
I was finishing my degree and then in Galway, in the west coast of Ireland, and thinking about what did I want to do? What was I going to do a PhD on if I was lucky enough to find one? And in third year, we'd done an essay on explaining the tuberculosis mortality rates in Ireland over the last 100 years.
In humans or in
humans?
Animals?
Yeah.
Okay.
And this was in the days before Google, so I went to my local library in Ballina, Mayo, one weekend and looked at some books and tried to find out about tuberculosis. Mm-hmm. And what interested me was, this was the late 1980s, and in the early 80s, the HIV/AIDS pandemic had struck, and you really got the feeling from that disease that it was this interplay of an infection, but also the social consequences of how that was seen and the stigma that was attached to it. And what struck me was the parallels with tuberculosis, a disease which in Ireland was still seen with, kind of alarm or fear. And then it's kind of awoken to me this idea that when we're studying infectious diseases as undergraduate students, we see them very sort of, I saw them, I should say, very kind of a one-track way. You get infected, you get disease, and that's it. But by looking into this essay, what really amazed me was how, your social status affected your probability of getting infected and also your probability of getting sick and dying. And so that was when, as well, in the early 90s, there was a big kind of hope around the ideas of molecular biology. So I decided, maybe I could do a PhD looking at the linkage of molecular genetics, trying to understand what makes bacteria kind of tick as pathogens. How do they work? And if you could understand the genes behind that, how does the genes in an organism equate to it becoming a dangerous bacteria? Well, then by understanding that, you'd have new knowledge that you could use to make new vaccines or new drugs to be able to combat TB. And here I am, 33 years later, I'm honestly still fascinated by that question. How do bacterial pathogens, how do any kind of germs or pathogens cause disease? It's both the inherent abilities of the bacteria itself and how it interacts with the host. And that can be true for a human pathogen like tuberculosis, but also an animal pathogen like the animal pathogens that cause tuberculosis. So it's really fascinated me always, that topic, yeah.
And your PhD was in UK or?
Yeah, so I
postdoc?
left. I had to leave Ireland and
sad.
went. Yeah, sad. I went to the University of Leicester to do my
Leicester,
PhD,
yeah. Mm-hmm.
and I was there for just over 3 years, and then I had a kind of a short fellowship that was funded by Wellcome. And I was lucky; my PhD had been funded by the Wellcome Trust as well in the UK, and then. As part of my PhD, like I said, I was interested in looking, how could you use molecular genetics to understand, new control strategies? So, for example, drug targets. So, my supervisor in Leicester, Professor Peter Andrew, was interested in cloning a gene that was responsible for susceptibility to a particular drug, isoniazid. And about basically halfway through my PhD, it was published by somebody else.
So you were scooped
I was scooped
badly.
I was scooped badly by a group of researchers at the Institut Pasteur led by Stuart Cole. So when I eventually finished my PhD, I thought if you can't beat them, join them. So I was lucky enough then to be able to start a postdoctoral research
in Pasteur Institute.
In the Institut Pasteur in France, yeah.
So you stayed in Paris?
I stayed in Paris for a while, yes. That was wonderful, yeah.
Yeah, they have one of the beacons in the field of microbiology and this type of molecular genetics work, isn't it?
Oh, yeah. It was a wonderful environment to...
With plenty of Nobel laureates.
Yeah, no, it's absolutely wonderful. And of course, it's the Institut Pasteur is in the heart of Paris. So you're in a wonderful research institute in one of, a global city like Paris. So I was incredibly lucky. But then as that was finishing, I always did think, I'd left Ireland, then went to the UK, then went to France. I was kind of moving eastward. I thought, I'm going to have to eventually, hopefully, get back home somehow to Ireland. And that's when I thought I needed to broaden my knowledge in terms of the animal pathogens for TB, so the bacteria that cause TB in animals. And that was when I decided then to start to try and work in the UK. So I went back to the UK and worked in basically an arm of the UK Scientific Civil Service, the Veterinary Laboratories Agency in Weybridge, which was, a global leading research institute for animal diseases, but also bovine tuberculosis, which is a problem in the UK and also a problem in Ireland. So I thought that by learning about bovine TB, I'd also then be able to, marry the two sides of human TB, bovine tuberculosis, and then eventually get back home, which I did eventually. Came back to Ireland in 2008. So you had a
complete tour, Tour de la France. That's great. It's so much of experience. Now, you started, as you said, more than 3 decades back. I think science was done in a pretty different way.
Yeah.
And pretty manual, pretty laboratory work. To right now, we are entering a phase where we are aiming that probably, or we are hoping that we don't need to do anything and AI will do everything for us. So you have seen all these transitions. Couple of things. Now, how much do you think our knowledge about tuberculosis has improved over this, complete evolution of this, what you have seen? And for example, I started my career, of course, in India, which carries a huge burden of the global pool of tuberculosis diseases or patients. In fact, it is kind of a big reservoir of a lot of different other mycobacterium species, including Mycobacterium leprosy, which was still a big problem when I started or when I entered the medical college in 1997. Now, this entire mycobacterium ecosystem, and of course, we have seen with our experience in India and also in certain sub-Saharan, subtropical tropical places as well, that body nutrition is a huge determinant. And of course, malnourished people are infected more and probably more with more virulence, or it affects more. So do you think nutrition is an important factor? And is it nutrition also because nutrition has a direct or indirect connection to body immunity status?
Yeah.
Do you think that body immunity status, which is kind of determined indirectly probably by body nutrition status. How much that is an effect, or how much is that much of a causal factor for tuberculosis? What is your experience?
Well, as you may know, there's been recently a very large clinical trial looking at exactly that question. If you take people who are infected with tuberculosis and change their diet, give them a good diet, what's the likelihood of them progressing to disease, or also just getting better quicker? And that's really been shown to be the case, that by improving people's nutrition, you can improve their status in terms of tuberculosis and making sure that they can fight off infection. And it's very kind of interesting because we think backwards into the 19th century, when there was the sanatorium movement, the idea of moving people out of kind of congested cities, bringing them into sanatoria, usually up mountains somewhere, improving their nutrition, improving their rest. And that was, before the days of antibiotics and other therapies, that was as good as it got, sanatoria treatment. And so we kind of almost like go back to the future to find our, things that we need to do to help combat TB. We need to treat it in a holistic manner. And I think sometimes for me as well, and I say this to the early career researchers as well, often, like I was saying to you, my view or my wish was always to use, we'll use these new tools of technology to dissect the bacteria, find its weak points, and from there develop these new therapies. So it's a very kind of a technological solution to the problem. But again, if you think back in the history of tuberculosis, there was a lot of kind of debate around that. Do you really need a technological solution to TB? Or do you just need to improve people's nutrition, their socioeconomic status? Do you need to improve people's general health? There was a very interesting study done, must be about 10 years ago now, by a group at the London School of Hygiene and Tropical Medicine with others, but showing that if you could get rid of extreme poverty and improve the social supports that patients need on tuberculosis, you could basically get rid of 85% of cases of tuberculosis globally. So you could really have a huge impact on TB without necessarily, we need new drugs and vaccines, but we also need to think that they're, one part, a kind of a technological solution. We also need to improve general health, general welfare of people, and ensure that we treat the infection in a holistic manner.
Do you think tuberculosis is then more of a public health problem than an infectious disease problem?
Well, of course, it's a huge public health problem.
Public health problem. Yeah. And I have seen that. I mean, because I entered medical college slowly but steadily. Of course, I got involved and kind of informed about the issue of tuberculosis. That was, of course, throughout Southeast Asia, not only in India. And then we were introduced to the different antibacterials, antimicrobials, including isoniazid, rifampicin, and that brought us immediately to certain other issues like resistance, cross-resistance, but I'll go back to that later. My first important, because you have kind of vacillated between both human and bovine tuberculosis. I have a couple of very interesting questions which I always wanted to ask you. Now, if I look into this typical, say, pulmonary tuberculosis, I'm not talking about disseminated infections, just pulmonary tuberculosis. And that same type of infection that you are finding in case of, say, in bovine cases, are they very similar in their evolution, or they have different symptoms? I mean, do they differ? I believe they are different species, so there will be certain differences, although the bacteria is probably the same, or some difference.
Yeah, so basically the major human TB pathogen is Mycobacterium tuberculosis. One of the first bacterial pathogens ever identified, and historically, noted on the 24th of March. 24th of March globally is World TB Day, and it commemorates the fact on the 24th of March, 1882, Robert Koch gave his speech at the Berlin Physiological Society. Yeah, when he says, this is it's not a hereditary disease, tuberculosis. It's an infectious disease, and it's caused by this bacteria. So that's Mycobacterium tuberculosis. Mycobacterium bovis is the dominant animal pathogen, and of course, as the name suggests, Mycobacterium bovis, it's the dominant cattle pathogen. And so from those two diseases, if we look at their pathology, so let's say, for example, we have a human case of tuberculosis, just from clinical presentation, you wouldn't be able to tell if it was Mycobacterium tuberculosis or Mycobacterium bovis that was causing.
So the X-ray, if you do the pulmonary X-ray, all the shadows, are there pretty similarity probably?
Yeah, consolidation. But as you said, maybe you're alluding to it, you're more likely to see extrapulmonary TB with Mycobacterium bovis, probably because of the route of infection, or maybe because the bacteria is just better able to disseminate once it's in the host. And there's lots of interesting kind of evolutionary and pathoadaptive kind of avenues that you can discuss down there.
Probably.
Yes, yeah. So that's the interesting thing. And I think sometimes as well, we have to think that's the problem in terms of when we talk about tuberculosis in humans, we always think about Mycobacterium tuberculosis and human-to-human transmission. Zoonotic transmission would be the movement of the infection from animals to humans, and that would be Mycobacterium bovis. But as I've said, how do you know what the amount is if the patient is presenting to you and, I don't know clinically, I can't tell the difference between the two bacteria causing the disease itself. And that the vast majority of tuberculosis in the world is diagnosed just on sputum. So people cough up a sputum sample. That sputum sample is stained and looked under a microscope for the presence of the bacteria. Down a microscope, it's very difficult to tell the difference between Mycobacterium bovis and Mycobacterium tuberculosis. So your ability to get good data on how much tuberculosis in humans is caused by Mycobacterium bovis, the data is very poor, but it's probably around 10%. So 10% of TB cases are caused by zoonotic transmission or Mycobacterium bovis, 90% caused by Mycobacterium tuberculosis, and that's in humans.
So you're saying that there is cross-contamination between species, interspecies cross-contamination?
Or transmission. Yeah,
but it's not unbelievably great. Or it's roughly, say, 10%, plus minus.
Yeah, 10%. Yeah,
give or take some
more. Yeah, but we do need good data at a global level. And, the other thing I should say as well is then it's the numbers when we're talking about tuberculosis. So when we're talking about the numbers of new cases, the incident rates of new cases of tuberculosis in humans year on year, that's roughly around 10 million new cases of clinical TB. Of that means that translates that about 1.3 million people die every year of tuberculosis. So
that's a significant number.
That's a really big number. And sometimes when we talk about these kind of numbers as well, I always find it 1.3 million. What does that actually mean? That means somebody dies of tuberculosis roughly every 25 seconds. This is a disease that we've known the causative agent, like I've said, one of the first bacteria ever, 1882, Robert Koch. Now it's 2024, 1.3 million people dying. And so that, I think, shows as well this kind of deadly synergy between poverty, low economic status, lack of access to healthcare, and then a pathogen which is very well adapted to infect and cause chronic infection and ultimately disease in human populations. And it really is that need to have technological solutions. We need better vaccines, better drugs, better diagnostics, but we also need to improve people's access to healthcare and improve their general living
nutrition
standards.
Status and other.
Exactly, yeah.
Now, of course, we cannot differentiate between Mycobacterium tuberculosis and Mycobacterium bovis under a microscope. But with this advent of huge technologies like RT-PCR, for example, all sorts of genomic analysis that we are able to do now in their labs, I think we can genetically differentiate with the genomic analysis between tuberculosis and Mycobacterium tuberculosis and Mycobacterium bovis.
Yeah.
How similar are they in a percentage scale?
Yeah, it's a great question. Yeah. So with the genomes of Mycobacterium tuberculosis and Mycobacterium bovis are both elucidated. So in fact, again, Mycobacterium tuberculosis would have been one of the first genomes
To be sequenced.
Yeah, published in 1998, again by Stuart Cole, and I was lucky enough to be in the lab at the time of that.
This time you were not scooped.
Yeah, this time I wasn't scooped. Yeah. And that was amazing. That really was amazing, because for my PhD, I had struggled to sequence 1,000 base pairs of a gene, a piece of sequence from Mycobacteria. And this was a collaboration that was done with the Institute Pasteur, many other labs, but a key collaborator was the Wellcome Trust Sanger Institute, the Sanger Center at the time. And so the Sanger Center had, of course, access to all the latest technology, sequencing technology, and we would get like 30,000 base pairs of sequence sent to us. Here's 30,000 sequences of DNA. What's in it? Tell me what's in this, or analyze it. And so that was one of the things that I was doing at Pasteur. And it was just amazing. After struggling so long to be able to sequence 1,000 base pairs, here I was getting these like data dumps of 30,000 base pairs. And it was so exciting to search and see what's in there, because you were looking at something that nobody else had ever seen before. You were trying to identify genes and therefore functions that, going back to my original thing of how does this bacteria cause infection and disease? How does it work? And you were basically being given the code. Well, here it is. You just need to translate it and figure out how it works. And that's what we were doing. Yeah. So if you look at then Mycobacterium bovis that was sequenced and published about 5 years later, the genomes are incredibly similar. They're about 99, over 99% identical. There's some chunks of DNA that are missing from Mycobacterium bovis that are present in M. tuberculosis, and point mutations, about 2,500 point mutations. So they're actually very closely related, very genetically similar, and yet they've adapted to different populations. Mycobacterium tuberculosis is a very well-adapted human pathogen. So that means it can infect, cause disease, transmit, find the next human host, and just keep on doing that cycle. And Mycobacterium bovis can do that as well in multiple animal populations. But, like I've said, Mycobacterium bovis can infect humans and cause disease. It doesn't seem to transmit very well, though, between human patients, but it can certainly infect us, and it can cause disease that's clinically indistinguishable from Mycobacterium tuberculosis. So that's why it's really important to break that zoonotic link, that transmission of infection from animals to humans. Because, I said about 10% of global cases of tuberculosis in humans are caused by zoonotic transmission in bovis. But that equates to, about over 100,000 cases a year, and over 10,000 deaths caused by Mycobacterium bovis zoonotic transmission. So to break that infection chain is why basically we try to detect animals that are infected, particularly cattle, and to keep M. bovis out of the food chain, identify animals that are infected, remove them from the food chain, and of course pasteurize milk to make sure that we get rid of any bacteria, pathogens that might be there.
Now, we are talking about this zoonotic disease. Of course, we are talking about the bovine population. From there can be transmission. There is another very interesting zoonotic transmission, I would say. That is the Mycobacterium avium complex, MAC. I mean, tell us a little bit about that. I mean, how difficult it is? How does it have a very poor prognosis? Does it only infect the lungs in humans, or is the manifestations always pulmonary, or are there kind of a more gross systemic manifestations?
Yeah.
And is it a bigger evil or a lesser evil or an equal evil?
Yeah, that's a great question as well. Yeah, so Mycobacterium avium complex is the group of bacteria that fall under that avium link.
Umbrella.
Yeah, exactly, umbrella. And as the name suggests, Mycobacterium avium. It's usually causing tuberculosis in bird species. But it's also a general kind of phrase for a lot of environmental mycobacteria. And certainly in people with maybe immunosuppression or issues there, you are, more likely to see disease in that population. So
opportunistic infections.
Exactly. Opportunistic infections, yeah. So we can think of Mycobacterium tuberculosis and Mycobacterium bovis. In fact, they fall under a group of bacteria, the Mycobacterium tuberculosis complex. We can think of them as more, I suppose, professional pathogens whose job it is, or they've evolved really to be able to infect, cause disease, transmit, and keep on going. Mycobacterium avium is probably the complex is a more environmental group of bacteria that are able as well to cause disease, though. And of course, again, if we think in cattle, for example, there's Mycobacterium avium paratuberculosis, which causes Johne's disease in cattle, which is a chronic enteritis, but that's in the gut rather than the lungs. So, they're a fascinating group and a very difficult, group of bacteria to work with. They're slow growing. I should have said that. So, what amazes me as well, if we think about the history of this, actually being able to identify pathogens, even in our optimized laboratory conditions, it takes us 2 to 3 weeks to get a colony to be able to see the bacteria growing on an agar plate from Mycobacterium bovis or tuberculosis. Mycobacterium avium, 2 to 3 days or so. But that's really still quite slow, and it shows how meticulous the original microbiologists who identified these pathogens were. They really were.
They really pinned it down.
Wow, absolutely amazing, yeah.
I know in many of these Southeast Asian countries where tuberculosis is still a huge problem, and there is a new resurgence given the antibiotic resistance that we are facing. I mean, I think BCG, for example, Bacillus calmette guerin, this vaccine has been used for a pretty long time. It works, but at the same time, we have to forget about eliminating or eradicating. We are seeing a resurgence in that. And BCG vaccine is part of national immunization programs in its different forms in different countries. Has it worked well, or if the vaccination, for example, smallpox, we have eradicated it. There are many other diseases where the incidence is decreasing because the vaccination is working. Even for COVID-19 pandemic, we saw vaccinations have worked. It can have certain side effects, but we are slowly understanding it. But it certainly has helped for us to get back to kind of a pre-pandemic lifestyle. But for tuberculosis somehow, despite integrating all these vaccinations in the national immunization program, which is compulsory in few cases, that results has not been very scintillating or something superly cherishable. So now my question is, why vaccination has not given us that kind of impact?
Yeah. So, it's interesting as well. If we think about BCG, so Bacillus calmette guerin, as you said, the only vaccine that we currently have available for tuberculosis in humans or animals, actually, in fact. So BCG, what is it? It's a strain of Mycobacterium bovis. It's the animal pathogen that basically Calmette and Guerin, two researchers at the Institut Pasteur in Lille, basically decided to follow the same kind of steps that Louis Pasteur had shown in terms of weakening pathogens, so attenuating them, making them weaker in terms of their virulence, and then using that weakened form of the pathogen as a way to inoculate and then generate immunity in humans that would then protect them from infection or disease. So BCG was first started to be cultured in the lab in around 1905, and basically they kept culturing it for years, like 13 years, kept on growing it. With problems in the between because of the First World War and stocks getting mislaid and whatever. But that's our only vaccine. The question is it a good vaccine? So it's a good vaccine in terms of a neonatal vaccine and stopping disseminated disease in children, so things like TB, meningitis, or extrapulmonary forms of the disease. So it's a very good vaccine that way. The issue is, what about in terms of an adult vaccine then? Something that's going to stop pulmonary transmission. There, the debate is not so good. And the protective efficacy is variable depending on which studies you look at and where it's being done. So, for example, when I was 9, 10 years old, I would have been vaccinated with BCG, basically repeating one of the first clinical trials that was done in the 1950s in UK school children, showing that if you vaccinated school children, they basically were protected 80% in terms of developing tuberculosis, so developing TB later on in life. So kind of protecting people as adolescents and young adults. However, if you look at studies done in India, so like in Shingleput, basically protective efficacy was 0. So you have a very wide spread in terms of protective efficacy, 0 to 80%. And it could be down to multiple reasons, the types of BCG strains that were used, the different exposures that people get during their lifetimes, how that impacts the ability of the vaccine to generate immunity. And then also I think as well, your point about smallpox, and, we show this up as, a fantastic example of when a vaccine works. But we've only eradicated one human disease and one animal disease, rinderpest. And we were able to do that because we had very good vaccines. We were able to have a vaccine that basically was able to block infection. And that's, of course, our holy grail. That's what we always want. We want a vaccine that's going to stop infection and give us like an invisible shield around us. But that's a very high bar to hit. So what's your kind of other option then? So as people know, well, in the COVID vaccine doesn't stop infection, but it does stop us getting, progressing towards disease. And that's going to be very important because disease can, obviously be debilitating, but also can be a way to stop, to kind of really potentiate transmission. So with disease, you're getting lots of bacteria, and so you're getting more chance of transmission because you've got more disease, more bugs, more transmission. BCG could be good at just maybe stopping that, transmission as well of pathogens. So it's good at stopping extrapulmonary forms of the disease, but we'd really want a vaccine that stops disease developing. And so again, BCG is basically a 100-year-old vaccine. So it's kind of tragic when you think of it that, again, one of the first bacterial pathogens ever identified, still with a huge case of mortality and morbidity globally. The only vaccine we've got is 100 years old. And again, I suppose the reasons for that is due to lack of investment in new vaccines, lack of interest, if you want to call it that way, in terms of developing new vaccines and the kind of market that they'd be used in. But there's hope because about 7 or 8 years ago now, there was a new subunit vaccine being trialed, M72, which is a fusion of 2 antigens of mycobacterium tuberculosis. That was formulated with a novel kind of adjuvant. And in 2019, that showed very promising results in terms of about a 50% efficacy at stopping progression towards disease.
That's hopeful.
Yeah, that's hopeful. And in fact, just in 2023, the green light was given for a phase 3 trial of that vaccine. So enlisting, over 25,000 patients who would be, or vaccinates who would be vaccinated. And then, you test then the protective efficacy. So how many people who are vaccinated, are protected against development of disease. So there's hope there, but it seems that we've been slow. Yeah, slow.
Just maybe a stupid technical question. I mean, BCG vaccine, you have said that it's made from attenuated strains of Mycobacterium bovis. Can't we make it from Mycobacterium tuberculosis? Isn't that would be great because it will be coming from humans?
Yeah.
What about that?
Yeah, that's a great question as well. Yeah. So in fact, there is So the one I spoke about there was a subunit vaccine, so taking a piece of the bacteria. There's another vaccine, which is in clinical trials, which is exactly what you say. It's a weakened form, an attenuated form of Mycobacterium tuberculosis. So now you've got all the antigens that Mycobacterium tuberculosis has, and that's called MTB-VAC. That's developed by a Spanish
I have
group.
heard about it. Yeah.
And that's rolling out to clinical trials as well. Yeah.
Again, let's hope for the best, isn't it?
Yes. So we just need more of those kind of avenues, more research to develop more of those potential leads, because as you know, you do end up getting dropout as you move along. Yeah.
Now, Steve, we can keep on talking about tuberculosis for days, and I think if we really get into vividly involved into it, we'll be still stuck within the studio for another week. That's not favorable. But in the last phase of this interaction with you, there are two things. A, I will request you that this is the season 1 of this talk, but I think there is so much to discuss. And I think a lot of physicians who are actually dealing with tuberculosis patients would love to hear more from you and to understand the wisdom that you have achieved over 33 years. So when we launch the season 2 of this talk, you are coming again.
Thanks.
That's number 1. And number 2, in this last few minutes, last phase of this interaction, I would like to collect your opinion about this resurgence of TB, where we already knew in the 1990s that we have these first-line drugs and second-line drugs, and isoniazid, rifampicin, ethambutol, pyrazinamide.
Mm-hmm.
But apparently, and that time already, they were given in regimes so that resistance in Mycobacterium tuberculosis or bovis against, if there is a microbe which is resistant against one drug, then the other one will still exert kind of a lethal impact.
Yeah.
And I have seen cases where regimes were used in a combination kind of a cocktail therapy of 4 to 5 anti-tubercular drugs. And still, resistance against tuberculosis has crept in. I think we talk less about it the mortality rate is very high, as you have said. And it's my opinion, though, that we don't highlight on this tuberculosis or hepatitis or these type of diseases, infectious diseases, as much as we should have done. We need better vaccines. We need more investment. Probably we need better infrastructure, better public health care system. Whatever be the faults, still there is a resurgence of tuberculosis. Mortality rates are increasing. And across all the age groups, tuberculosis is wreaking a havoc. I will quickly like to understand this. What drives this resistance? Why Mycobacterium tuberculosis? I know it's evolutionary-wise very well adapted microbes. So it knows the rules of the game. But still, for lack of a better word, our failure.
Yeah.
That has been quite depressing and frustrating. Why is that?
Yeah, again, it's a great question because, if we go back and think about, treatment of tuberculosis, again, one of the first antibiotics, streptomycin, developed by Schatz and Waxman, was one of the first antibiotics identified. And as you say...
They got Nobel Prize also for that.
Yeah,
yes.
Waxman got a Nobel Prize.
And there was some controversy, but let's not get there.
Exactly. That's for the next podcast.
That's for a separate episode, yes.
That we think, you think about it actually, tuberculosis was one of the first infections where that idea that you just mentioned, combinatorial treatments, was brought to the fore because they realized that with a single drug, you were getting relapses. So you needed to combine drugs to reduce that relapse rate. And so, we ended up with, as you said, with the introduction of rifampicin in the late 1960s, 4 drugs, 6 months. So basically 4 drugs, 2 months, and then continue on with rifampicin and isoniazid for another 4 months. So a 6-month drug regime, which works. But the drugs are not, they're pretty toxic and they're not the nicest drugs to take. So can you blame people who, once you start to feel a bit better, your bacterial load drops, compliance can be an issue.
It is an issue.
Yeah,
in fact,
absolutely.
That is why before leaving India, I saw that, and I'm sure it has been replicated in many other countries, that compliance is such a big issue that they started DOT therapy, directly observed therapy. and excellent health workers, very dedicated. They were sent to home so that they observe that patients are actually taking these medications.
Yeah.
Because as you said, the cocktail therapy is not a great stuff. You're taking a lot of toxic materials inside your body, and you might end up suffering from something else from that treatment rather than the disease itself.
Yeah. So I suppose I should say the side effects. Yeah, the side effects of it.
Side
So you
effects
are kind of balancing the two things. And so, I think from the 90s onwards, like you say, there was this refocus with the emergence of drug-resistant tuberculosis, the reality thinking of, we need new drugs. And that has happened. So, rifampicin was the first drug, the 1960s, sorry, I should have said, the last kind of drug that was added into that regime. And then in the early 2010s, you had bedaquiline appearing, almost though 50 years later, as the first FDA new kind of drug. But you've got other ones. You've got delamanid, pretonamid. So you do have new options, and those new options are showing great promise, because the problem with multi-drug resistant tuberculosis is if your frontline drugs, your 6-month therapy, your kind of best shot, doesn't work, well, now you're going to be on to 18-month therapy with more second-line drugs, which again have more severe side effects, going backwards to things like streptomycin or injectables. Now, because of the new drugs that have been brought online, the WHO last year or the year before released recommendations for this new regime, so even for drug-resistant tuberculosis, with these new drugs, things like bedaquiline, you can now have a 6-month therapy for multi-drug resistant or rifampicin-resistant tuberculosis. So this, again, I think shows we're desperately needing more research and development to develop these leads. But also, once you have the leads, rather than taking multiple pills a day, what if you could just take a single pill? You think going backwards, again, I think of when I started talking about this, HIV, how HIV and therapy has changed from, 1980s up to now, from multiple drugs being developed, rational drug design, and now to developing, single pills that have multiple combinatorial treatments within them. So making compliance easier, because you're making it easier for patients to take their drugs. So I think that's what we desperately need for tuberculosis, but also lots of other chronic infectious diseases.
Do you see that type of drives being taken in Ireland or UK per se, because you have a lot of experience in the United Kingdom? Do you think this type of drives are being made, or because they're very expensive and non-profitable, sometimes taken?
Yeah, I think that is an issue, yes. It's like what pharmaceutical companies are interested in developing new antibiotics. It's an issue for tuberculosis and lots of bacterial infections in general. And as we know, we're therefore being caught in this, I suppose, that kind of unseen or pandemic, as we talk about the antimicrobial resistance. We're really not taking care of the antibiotics we have at the moment well enough as we should be, and we're not developing new ones. And I think that disconnect between the need to develop new drugs, but also, of course, the need for industry to see as though they're making a profit, can be very difficult. And of course, that's where, again, if I talk about the vaccine, the M72 vaccine, that was, first phase 1 and phase 2 clinical trials of that were done by GSK. But now that next phase 3 trial is being funded by the Bill and Melinda Gates Foundation, because of the huge step up in terms of funding that you need for it. So we are caught in terms of, that model of developing drugs and making a profit from them, which is, I can understand, but also the need for, we need other ways to develop public-private partnerships or state-based interventions which keep these drugs, the pipeline kind of flowing for new ideas, but also to make sure that those new drugs are then available.
Very recently, somewhere I read a very funny thing, that quote, that modern humans are the only species which is risking its elimination because it's financially not profitable. Sometimes I believe probably because the way we are, I had Gerald Barry in my last episode, and it's fascinating, and I learned a lot of things from these interactions so far. And apparently most of our response to COVID was pretty similar to that of Spanish flu. that's what I learned from there as well. So it's sometimes interesting that whether we are evolving forward, backward, and apparently we are even willing to risk our destruction, extinction, because it's financially always not very profitable.
Yeah.
It's an interesting lesson for all of us. Last question. Imagine for a moment, hypothetically still, you have all the power in our bureaucratic system, political system, to make changes in public health care. Or you have to advise that these are the 5 things or 4 things, I don't know, that you should do immediately, or we should install immediately, that will at least give us some relief from this menace of tuberculosis. You have all the power. I know you will never join politics, but still, if you have all the powers, what are the very few pearls of wisdom you will immediately throw to the AGM?
Yeah, it's a great question, because again, what would you actually do in an ideal world? And I think, in some ways, it's that kind of a balance between the technological solution and the kind of social context that you're offering that solution in. So I think you've got to improve access to healthcare across global populations. You've got to be able to diagnose infection to be able to treat it. So you've got to be able to get people accessing healthcare, getting them diagnosed, and then getting them the proper treatments. And the proper treatments, like I said, our drug regimes are decades old. Our vaccine is 100 years old. We need those technological solutions to come online much faster than they're currently coming online, to be able to, give us more options for treatment and also prophylactic prevention in terms of vaccines. But ultimately, I think you've got to be able to make sure that the people who need the vaccines, the treatment, and to be diagnosed with infection can get access to that healthcare, free at the point of access. So for me, that's what I think. So it's balancing up those 2 sides of the coin, the social aspects of infectious disease and public health, and those, going back to my naive, innocent young self, the technological aspects of what can we do with the technology that we're developing to develop newer solutions rather than the older current things that we're depending on.
I think what I also distill out from what you have said, that you will certainly invest more in strengthening our public healthcare system. Yeah. Which is, again, might not always be financially very profitable. So you have to convince a lot of people, I believe. But I think that's a very good cause. That's a very good kind of a, not solution, but at least in a way to address these things. And I think these issues are being reverberated or being reiterated, and it's getting resonated more into the policy making circles. And I'm quite certain, at least in this comment, you will not be scooped. So, Steve, thanks a lot. It has been kind of a more relaxed atmosphere that I had chatted with you. There are zillions more questions, and I'm sure it's because this podcast is, I am seeing there are audience more and more joining from different parts of the world.
Yeah.
And I think there is a great interest in tuberculosis. It's a very old, archaic disease known from very early days, and newer things are arriving. So it's kind of getting slightly out of focus, and I think that's not should be the case. So you're coming back in season 2. I'm already booking you out, and we will keep on discussing where we are leaving today, unfortunately, because of the time crunch.
No, that's a pleasure.
But it's my absolute pleasure. Thanks to you. Thanks to all our technicians who have worked very hard for this podcast to be what it is getting there. And what can I say? We'll meet tomorrow in UCD.
Thanks, sir. It's a pleasure, honestly.
Thank you. Thanks a lot.
Thanks very much.